It's my great pleasure to be here to talk about this topic. So, yeah, we're going to talk about LAG-3 for cancer. And this is the molecules really beyond PD-1. As you know, PD-1 has been successful in cancer immunotherapy. And then LAG-3 has become the number three checkpoint receptors, has been showing some efficacy in cancers. So this is a very different molecules. LAG-3 is very big as CD4. I would say LAG-3 is four times bigger for the ectodomain compared to PD-1. So this field is established by Fedor Tribel. Tribel started a company called Immunotherapy, and Dario Vignali. So this molecule was discovered almost as old as PD-1, almost 30 years ago. As I said, so CD4 homolog, class 2 as a canonic ligand, so that's why class 2 binds to TCR, CD4 actually same time. And then this is inhibitor as similar to PD-1 suppress T3 responses, and then you can also suppress CD8 T cells. So that's why there are some other mechanism has been suggested because class 2 normally associated with CD4. So how class 2 suppress CD8, that has been a debate. So that's why early time I spent quite a lot of years to understand what might be the possible ways for suppressing T cells, especially CD8 T cells through LAG-3. So there is some mechanisms, for example, FGL1 I discovered potentially control CD8 T cell responses. So because of this, right? So there are several different ligand, and then this is molecule suppress T cell responses. So that's why very similarly, people has been generating antibody to block these pathways. The first antibody was generated by BMS, relatlimab. Actually, BMS has been working with Matrix, acquired Matrix. They have generated nivolumab, PD-1, relatlimab, LAG-3, and also CTLA-4, ipilimumab. Three molecules generated by BMS all become the drug. But however, relatlimab did not show single agent activities. This antibody block both ligand FGL1 class 2, and recently, although relatlimab did not show single agent activities, but the combination with nivolumab is called Opdualag. This is a frontline therapy, already become frontline therapy for melanoma because I'm going to show you some data. So this is the first combinational drug. It's not combination drug. They just made a manufacture of two antibody together. It's a fixed dose, 400 mg PD-1 and 160 mg LAG-3 antibodies. So, manufacturer, like become the core manufacturer, and then already become the frontline therapy for melanoma immunotherapies. So one slide. So the original data from melanoma from that was 2022. The New England Journal, they reported the comparison of nivolumab versus relatlimab nivolumab, this combination, the combo. So the PFS, if you look at this data, PFS data was like p-value is about 0.006. That was the major data leading to the relatlimab nivolumab combo approval. So as I said, so the combination was like nivolumab 480 mg, and relatlimab was very little, 160 mg. I think that at that time they were talking about because BMS have seen some toxicity of nivolumab combination with ipilimumab. That's why they were a little bit reluctant to start with a very higher dose. So that's why they start with a very little LAG-3, 160 milligram fixed dose. So p-value was okay, but the later time reported OS data was not very significant. So that's why the PFS data generating the most of the confidence leading to the approval of this compilation for melanoma. That was almost 4 years ago. Can you move forward? Thank you. So a later time, they update the data. So PFS data, as as predicted, hazard ratio is about 0.78. But they also later time, summarize the OS data, overall survival. P-value was about 0.059. I think it's close, but it's not very significant. So that's why for the long time, BMS has been focused on the PFS. All right, one more slide. So recently, as you know, nivolumab and ipilimumab PD-1 CTLA-4 combo has been proved for melanoma. So one question here is how about PD-1 LAG-3 combo versus PD-1 CTLA-4 combo? So this is the data just summarized about 1.5 years ago. So they compare both PFS and OS. The data looks interesting that both combination, either PFS or OS, data is very similar. There's no significance, no difference, not too much difference at all. But however, for the adverse effect, if you talk about grade 3 and 4, so relatlimab and the nivolumab combo, PD-1 LAG-3 combo is basically like 30% of PD-1 CTL4 combo. So this data is very exciting because that means safety is much lower, 30%, but however, efficacy is very similar to PD-1 CTL4. Because of this, and also the any grade treatment related adverse effect is also leading to discontinuation is also much lower, 17% versus 41%. So all this data leading to a very exciting direction that PD-1 CTL4 combo might be better than PD-1 PD-1 LAG-3 combo might be better than PD-1 CTL4 combo because of safety. Efficacy might be similar. So that's why this um, becomes the basis of relatlimab, nivolumab as a frontline therapy for the melanoma. And also, this give other programs from other companies more confidence to show whether their program might be better or better design, can lead into approvals in melanoma or beyond melanoma. One more slide. That's right here. So this is a summary. So LAG-3 PD-1 combination versus PD-1 CTL4 combination. The efficacy looks similar, but toxicity is much lower, and also discontinuation rate is much lower. So this is also the why this program is getting exciting at BMS. One more slide. So then, BMS is becoming the number one companies, especially in the LAG-3 field for cancer immunotherapy. But many other company also has been working on LAG-3, but unfortunately, many companies discontinue those programs because they didn't see single agent activities. If you want to push forward, you have to really combine with PD-1. So then you have to prove whether your antibody is better than BMS or your program design is better. So that's why Regeneron looking into this question, they want to think about maybe their potential, their program have some potential. For example, their antibody actually have a little bit different epitope. And then they also look into the question that because this program is safe, why not we have a much higher dose of LAG-3 on top of PD-1? So that's why they want to have better designs. Not just dosing, number one. Second is maybe clinical trial designs. So they start a trial. I think melanoma is no hanging fruit. They want to start a trial to compare. So for example, for BMS, as I said, nivolumab for 480 milligram and the relatlimab 160 milligram fixed dose. They want to start with much higher LAG-3 antibodies. For example, they use 10 times more, 10 times, the dose as of relatlimab. For example, they use 16 mg, 100 mg, 1,600 mg, 10 times of relatlimab, and compared to versus, and also they use their own PD-1 antibodies, semi-pilimab. This is a higher dose combination. They also want to see whether dose is very important. So that's why they also have another antibody, sorry, the other design, which is 400 mg LAG-3 compared to the same in combo with the same dose of PD-1. And also their dosing is a little bit different every 3 weeks. For retinamides every 4 weeks. As a comparison, they want to potentially beat Keytruda, pembrolizumab, and also they have another group which is their PD-1. So the idea is whether they want to see whether there is a dose response of LAG-3, number one. And second is whether this combination is better than Keytruda. And of course, you also want to see whether their PD-1 program is similar to Keytruda, but that's a little question because they already have some data showing very similar efficacy of their program with Keytruda. So then key question here is, are there any dose response? And the second is this LAG-3 combo better than PD-1, especially better than Keytruda? So then data here is, they just released the data two weeks ago. So as you can see here, the four group, each group have, especially for LAG-3 combo, they have 400, 500 people. And Keytruda is also have 462 monotherapy. So as you can see, the primary endpoint is PFS. There is clearly a dose response, especially 500 patients for the high dose versus low dose 400 patients. So, PFS um, median PFS is 11.5 months versus lower dose is 5.6, and compared to the pembro is only 6.4. But the p-value, if you look at the p-value, only high dose showing close to significant 0.06, but the lower dose basically very little difference. So strategically and also statistically, I think the trial was failed because a major PFS, median PFS endpoint did not met significance. That's bring the disappointment to the field. But to me, I think the data is very similar to BMS, but the p-value is very close, but it's not very significant. One more slide. So then the summary is the melanoma trial did not reach to statistical significance for its primary endpoint of PFS. OS data is getting, is not ready, but because this is a primary endpoint, that's why to conclude, at least at this point, is not significant. But there is clearly a dose response. And then the setback is very close. And another good thing is no new safety signal. This data also similar to BMS. So that's why to summarize, I think actually did help, but the problem here is there is some kind of statistical analysis, and even OS data will be, let's say if OS data is getting interesting, but still you might need to run another trial. So that's why I think all this data is quite similar to BMS, but the problem is, they didn't reach to the significance. But I do think there is some kind of potential analysis might generate something interesting. So for example, the OS data, as I mentioned, and the second is there are some other Regeneron trials. They also have another trial directly compared with BMS, relatlimab, nivolumab combo. Um, Whether that can be successful, I don't know, but let's see whether there's any good news. So other Regeneron trial, they also have a trial in lung cancer. Unfortunately, that was also failed last month. So that's why I think in general this is a negative trial, but I think there's some hope. For example, patient stratification, potential biomarker in the tissue or in the blood. But even this biomarker might generate some significance, but all of those require a lot of trial. So the question then is how confident Regeneron is to start another trial because they already have 500 patients each arms. This is 1,500 patients, big trial. If we have something interesting, is Regeneron confident enough to run a lot of big trials? So that's why all of those data I mentioned about BMS, I mentioned about Regeneron, all of those data pointing out that this is a very interesting pathway, but this checkpoint receptor ligand biology is still interesting, is a key. Some of those are not very clear, but I think someday for the LAG-3 field or even beyond LAG-3 field, there will be something interesting. There will be some interesting news. So I don't know. I think Regeneron are going to make some good big decision in soon whether they want to get a better analysis or start a new trials or finalizing the trial compilation or direct comparison with relatlimab. I think there will be something interesting coming out, but let's see. Hopefully next time we can talk about that. So at the same time, I think because of those setback and failure, Regeneron also have some strategic thinking. So that's why I'm going to mention about the same news from Regeneron. They want to start another direction on slides. So Regeneron at the same time as during setback, so they announced strategic collaboration with a company called Parabilis Medicine. This is a very interesting company. They have a very novel antibody-helicon conjugate. As you know, recently because of setback in IO, many companies jump into ADC area or antibody plus area, PD-1L2 or PD-1 VEGF or PD-1 ADC or even PD-L1 ADCs or antibody conjugates for tumor antigen stuff. But this company is doing something very different. So Regeneron have started a collaboration with Parabilis. The first front payment is about 150 million, 125 million, and also some upfront, maybe like 200 million in total something, actually more than that, plus equity commitment. So, and the long term, maybe $2.2 billion. So this is a very big commitment and also quite interesting directions. So what is this antibody helicon conjugate? One slide. So most of the company focusing on antibody conjugate are conjugating, for example, small molecules like toxin to kill tumor cells. Or recently there is AOC, RNA or nucleotide conjugation that is called AOC. ADC is a drug, right? Like small molecule something. So this is a helicon conjugate. So what is AHC? So basically, it's antibody conjugate with a peptide. The peptide is alpha helix and quite stable. It's a kind of peptide antibody conjugate. So then this helix, they can have some stability, drug-like stability on one side. Second side is they have some cell permeable property to make this antibody trafficking to the cytosol. But I would say traffic endocytosis is not very hard, but how to escape from endosome whatever to the cytosol, that is a headache. So you have to screen something very stable as put as drug like as possible, and also better to mediate endocytosis, and also the drug has to escape from the endosome. That is something also not very easy. But interestingly, they already proved concept. For example, their first programs is the peptides can inhibit beta-catenin with TCF4. The peptide get inside will block that pathway, so they already have proof concept. So if that can work, you can have a peptide get into cytosol to block many cell interactions. So this is very different from PROTAC or from the AOC, or you have to silence a gene program something. It is really peptide driven, so then you can have many targets to be targeted by this type of approach. So I think that's very interesting to the field. I still don't know, the whole field still don't know how useful, how much potential this program is. But based on the interest from Regeneron, we really want to start a new direction. Right, one slide. So this is also the summary. They have four different type programs, either in the phase one, phase two. The first program is already phase two. They already planned phase three. It's the same drug, but they have very different directions, different focus on cancers. They also have several different programs targeting cancers. So that's why I think this is a very interesting direction that Regeneron have some failure in LAG-3. Of course, they still have some trial ongoing. But the second is they start new direction to exploratory. This is also a reason why I like Regeneron. is very exploratory and very ambitious. I would like to wish their good luck for both directions.